Welcome everyone today to this year's Lasker Awards ceremony and a celebration of individuals who have made outstanding contributions to science. I'm Mike Overlock, and I have the privilege of being the chairman of the Albert and Mary Lasker Foundation. Today marks the 69th anniversary of these most prestigious awards. As in years past, today we are recognizing groundbreaking science central to the Lasker Foundation's mission of improving human health by accelerating support for medical research. As is our tradition, I would like to acknowledge my fellow board members of the Lasker Foundation who have so capably guided the Foundation's approach to its mission. Would the members of the Board of Directors of the Lasker Foundation please stand and be recognized? Thank you. Every year, this luncheon is graced by the attendance of previous Lasker laureates, whose work enables the development of tomorrow's therapies and cures. I'd like to ask the Lasker laureates to stand and be recognized. Thank you. In your program on page 7 is a list of our dedicated and insightful awards jury, ably chaired by Joel Goldstein, who we'll hear from shortly. This is a unique group of talented people whose work identifies the awards winners from a group of highly competitive, dedicated, and talented medical researchers. We are grateful for your service. Would the members of the awards jury please stand and be recognized? Thank you. The jury selected an impressive group of awardees this year, and we have gathered to honor them. Doctors Morris and Walters are here to receive the Albert Lasker Basic Medical Research Award for their discoveries concerning the unfolded protein response. Doctors Benabid and DeLong are the recipients of the Lasker DeBakey Clinical Medical Research Award for the development of deep brain stimulation for treatment of motor symptoms of Parkinson's disease. And Mary Claire King is today's honoree of the Lasker Cosland Special Achievement Award for her contribution to genetics and human rights. A round of applause. You'll hear more about their accomplishments of our winners later in the program, but now I would like to ask Claire Pomeroy, the president of the Lasker Foundation, to say a few words. Thank you very much.
Well, thank you, Mike, and welcome, everybody. One of my most important jobs is to encourage you to start eating your salads, so please enjoy. It is so wonderful to join with all of you today to celebrate medical science and to celebrate our amazing winners. I am truly honored to serve as president of the Lasker Foundation. You know, I'm energized every day by the Foundation's mission to improve health by accelerating support for medical research, and we do that by recognizing excellence in research. We educate to raise awareness, and we advocate for sustained funding for medical research. The dedication with which Mary Lasker enlisted her fellow citizens in her decades-long crusade to increase funding for medical research is inspiring. Indeed, the fact that we have so many brilliant and wonderful people here today is a testament to her vision. Unfortunately, At this moment in history in the United States, that vision is severely challenged. Flattening of the NIH budget over the past 10 years has led to a 20% loss in purchasing power for medical research over the past 10 years, calling into question whether the types of advances in basic understanding of biology and the development of innovative clinical techniques that we celebrate today, if we'll have that foundation to build on so that we can celebrate them tomorrow. We must not let that happen. We must all contribute to work to ensure sustained and sufficient funding for the NIH, the NSF, and other federal grant programs. And I believe we must also expand innovative approaches to funding that bring together government and industry and academia and philanthropy so that we all work together to ensure that our research enterprise finds the answers we need to create a healthier world. You know, it has been said right down at the United Nations, never for the sake of peace and quiet, deny your own experience or convictions. And I call upon everyone here not to deny their convictions about the importance of medical research. We can justifiably and proudly celebrate the fruits of the investments we've made in the past, but we cannot abide complacency. We must join together as scientists, as citizens, as policymakers to ensure that we invest in our future, that we are able to benefit from exciting triumphs to come. So I would like to thank the Board of Directors, our wonderful board chair, Mike Overlock, for their dedication to the Lasker Foundation. I'd like to thank each of you for being here today and for supporting this cause. And a special thanks to Joe Goldstein, who so capably, as we heard, guides the Medical Research Awards jury and who cares deeply and passionately about the future of science. So Joe now has a story to tell us. It's a story about misbehavior. I'm looking forward to it. Joe Goldstein.
Thank you, Claire. 125 years ago, the Irish playwright Oscar Wilde famously proclaimed that the most creative individuals are those who have taught their minds to misbehave. And the great virtue of a misbehaving mind is that it endows one with the ability to spot the next thing, whether it be in art or in science. And the 19th century novelist Honoré de Balzac passes Oscar Wilde's misbehavior test with flying colors. In the 1830s, Balzac conceived the idea of describing the grand sweep of Parisian society in all its splendor and squalor, from the highest aristocrats and politicians to the lowest swindlers and prostitutes. And over a 16-year period, he published 91 novels and short stories, and shortly before his death in 1850, he organized his 91 works into a multi-volume collection called The Human Comedy. Balzac's writings contain many astute insights into human behavior, and one of the most original and a prime example of his misbehaving was his analysis of how certain people get ahead in life, not by hard work, but through advantageous marriages. So Balzac is responsible for a major literary innovation that has stood the test of times for nearly 2 centuries. He invented the device of serialization, in which the same characters appeared over and over again in his different books. And serialization has since been adopted by hundreds of writers, and without serialization, J.K. Rowling would not be a billionaire with wealth exceeding that of Queen Elizabeth. And serialization also accounts for the great popularity of TV programs such as Downton Abbey and House of Cards. Well, 50 years after Balzac's death, the French government commissioned the sculptor Auguste Rodin to create a statue in memory of Balzac, whose work by that time had achieved international acclaim. Rather than producing a realistic physical likeness of his subject, Rodin decided to do a little misbehaving and break with the sculptural traditions of the past. He shocked the Parisian establishment by creating a semi-abstract figure in which only Balzac's head remains visible, with the rest of the body wrapped in the dressing gown that he wore when writing. Monument to Balzac, completed in 1898, is considered the first truly modern sculpture. A prominent art critic of the time described the sculpture in the following way, end quote, "Balzac stands with his huge head thrown back, alert like a wild animal, drinking in with eyes, nostrils, and lips, and scenting the fever of the human comedy," end of quote. Bronze cast of Rodin's Balzac are displayed at more than 50 locations throughout the world, including 2 museums here in New York City, MoMA and the Met. It is one of the 2 or 3 most viewed sculptures in the world. One of Balzac's intriguing stories, entitled The Unknown Masterpiece, deals with the subject of creativity at the most fundamental level. It tells the tale of an artist ahead of his time. Unknown Masterpiece was first published as a short story in 1831 and later integrated into the human comedy. The story is set in 17th century Paris in a studio on the left bank. The plot is simple. A famous aging artist named Frenhover is obsessed with finishing a painting he has been secretly working on for 10 years, and one that he claims will be his masterpiece. A painting that portrays a beautiful nude woman with such brilliance and artistic skill that she seems to be actually alive. When the 2 younger painters, who are great admirers of Frenhover's work, persuade Frenhover to let them see the secret canvas, they are appalled and shocked. All they see is an indecipherable jumble of strange lines in paint. As they stare in horror at the work, they mock the older artists and conclude that their celebrated hero has gone mad. Realizing his failure, Frenhover abruptly bids farewell to his 2 friends, burns his painting, and mysteriously dies during the night. The tragedy of Frenhover and the brilliance of Balzac is that the fictional artist had created the first abstract painting, inventing abstract expressionism 125 years before Jackson Pollock. The crisis that Frenhover faced in his failed quest for artistic perfection exerted a profound effect on Picasso, whose paintings were so far ahead of their time that few of his contemporaries recognized them as masterpieces. In 1931, for the 100th anniversary reprint of Balzac's Unknown Masterpiece, Picasso produced 13 etchings to be included in the special edition. And like Frenhover's painting, Picasso's drawings were kept a closely guarded secret. And when they first appeared in the centenary book, some of them looked like a mass of lines and smudges of ink, such as the one shown on this slide. The jumble of lines here is typical of Picasso misbehaving. Does the jumble represent the yarn of wool in the model's hand, or is it Frenhover's abstract painting? Today, Picasso's 13 Balzac etchings are considered landmarks in the history of engraving. When a journalist asked Picasso, "What is the secret to your genius?" he remarked, "When I was a child, I could draw like Raphael, but it took me a lifetime to draw like a child." Picasso was so haunted by the ghost of Balzac and the tragedy of Frenhover that in 1936 he moved his studio to a townhouse located at the exact same building in which the setting for the Unknown Masterpiece took place 100 years earlier. It was at this address that Picasso painted his greatest masterpiece, Guernica. Soon after Picasso moved into his new studio, German warplanes bombed the Spanish city of Guernica, and Picasso devoted full energy to his one large canvas, which he completed in record time of 3 months. At the unveiling in 1937 at the Paris Exposition, Guernica perplexed the critics. But unlike Frenhover, the imperturbable Picasso was unfazed by the negative criticism, and he lived to see his work evolve from an unknown masterpiece to a highly celebrated one. 4 years ago, in 2010, the National Gallery of London invited the British painter Richard Hamilton to stage an exhibit of his most recent work. At the time, Hamilton was 88 years of age and still exceptionally productive. Like Picasso, Hamilton had come under the spell of Balzac's unknown masterpiece, and the National Gallery's invitation provided him an opportunity to express his ideas on the search for artistic perfection. A brief background on Richard Hamilton will help those of you not familiar with his career to appreciate how he responded to Balzac's unknown masterpiece. Hamilton is one of the most influential British artists in the 20th century. He is widely regarded as the founder of the pop art movement in the 1950s, predating Andy Warhol and Roy Lichtenstein by several years. One of Hamilton's iconic works was a 1956 collage entitled "Just What Is It That Makes Today's Home So Different, So Appealing?", in which he used images cut from American mass circulation magazines of the 1950s. The collage depicts a contemporary Adam and Eve, a muscle man holding a giant lollipop, and a nude woman on a sofa with a lampshade on her head. And Adam and Eve are enjoying their living room filled with all sorts of new American consumables: a tape recorder, a television, a vacuum cleaner, a canned ham on the coffee table, and a brand new comic-style painting on the wall. According to the art critic John Russell, with this one collage, Richard Hamilton, quote, single-handedly laid down the terms within which pop art was to operate, end of quote. The first time the word pop ever appeared in a painting was when it was emblazoned on the lollipop at the center of Hamilton's collage. Now, if anyone had a misbehaving mind in the Oscar Wilde sense, it is Richard Hamilton, the epitome of British pluck and originality. Over a 60-year career, he innovated, experimented, reinvented himself, and spotted the next big thing before any of his contemporaries. At age 80, Hamilton taught himself computer graphics. Now, one of his most politically provocative computer-generated paintings is entitled Shock and Awe. Here we see life-size Tony Blair dressed in a pistol-carrying cowboy costume presiding over the invasion of Iraq. And one shudders to think how Hamilton might have satirized George W. Now, once Hamilton had mastered digital technology, he devoted his full energy, beginning in early 2010, to the National Gallery's project, his homage to Balzac's unknown masterpiece. Using computer graphics, Hamilton composed a montage of 4 known images that he assembled digitally and refined with Photoshop software. Nothing was painted. The centerpiece of this montage is a reclining nude representing Fraunhofer's Muse. Hamilton created this image from a digital scan of a photograph of a 19th century nude whose pose harks back to Titian's famous Venus of Urbino. The nude is surrounded by 3 famous artists from left to right: Poussin, Courbet, and Titian. Titian represents Fraunhofer. Poussin and Courbet represent Fraunhofer's 2 younger artist friends. The 3 artists were scanned digitally from reproductions of their own self-portraits. The 3 artists are all absorbed in deep thought, perplexed by the mysterious sensuality of the nude. A notable feature of the montage is the empty easel that is centrally placed between Poussin and Courbet. The H-shaped easel is identical to the one that Hamilton used in his studio and may be meant to represent the specter of Hamilton himself. The reclining nude is perfectly shaped, but she appears unreal in her glossy skin, digitally exact but artistically artificial. Within days after completing the digital montage, Hamilton suddenly died on the eve of his 90th birthday. His original intention was to use the digital montage as a guide for creating an authentic oil painting, which he believed would capture Fraunhofer's ideal of living purity that is missing in this pixelated version. As fate would have it, Hamilton's masterpiece, like that of Fraunhofer's, will remain forever an unknown masterpiece. a bittersweet irony if there ever were one. The empty H-shaped easel in the montage now takes on an eerie meaning that the misbehaving mind of Richard Hamilton may or may not have intended. We will never know. The misbehaving minds of Balzac, Frenhofer, Rodin, Picasso, and Hamilton endowed each of them with the uncanny ability to spot the next big thing before anyone else. Spotting the next big thing is a distinguishing characteristic of scientists who win Lasker Awards and Nobel Prizes. And after lunch, you will hear how this year's Lasker winners were the first to spot how cells handle unfolded proteins, how the tremors of Parkinson's disease can be alleviated, and how breast cancer, certain forms of breast cancer, can be caused by an inherited gene. And now it's time for all of us to join our Lasker laureates at spotting the real next big thing, our food and wine. Enjoy your lunch.
So thank you, Joe. The one misbehaving mind I may never be able to decipher is yours, so maybe that will be next year's lecture. Please enjoy your lunch, and after the main course, when dessert is served, we'll come back to introduce our winners. Thank you very much, and welcome again.
The first award that will be presented is the Basic Science Award, and the presenter will be Mike Brown. Mike Brown and I have actually worked together for 43 years. Can you believe that? We're the longest partnership in science. And Mike has been on the Lasker jury for 18 years. He's been on this podium a number of times, and let's hear what he has to say about the Basic Award. Mike?
You know, Joe is right. I've been to quite a number of these, and I always, you know, I was thinking just now, you know, they introduced the board, and of course we have a wonderful board. And then they introduced the jury, and we have a wonderful jury and our brilliant chairman. But I think what makes this award special is the audience. I think to come to New York and see this amazing gathering of scientists, academic leaders, industrial leaders, political leaders, to come here to honor biomedical science, I think it's a fantastic tribute to New York, to be honest. Okay, my job is to present the Lasker Basic Science Award. So Henry Ford is credited with inventing the assembly line. But Mother Nature beat him by 1 billion years when she invented the endoplasmic reticulum. Mother Nature's assembly line. In Ford's assembly line, parts are passed from one worker to another. At the end of the line is the inspector who tests the product. Correctly assembled products are exported and defective products are destroyed. If too many products are defective, the inspector sends a signal to the control room where supervisors dispatch troubleshooters to correct the problem. Ford never knew he was copying the endoplasmic reticulum, which I will abbreviate as ER. The ER is a network of membrane-bound tunnels within every nucleated cell from yeast to humans. Like Ford's assembly line, the ER makes products for export. Its products are proteins designed to function not in the controlled environment of the cell, but in the harsh environment outside. Some of the proteins are destined to reside on the cell's surface, where they serve as receptors that respond to external signals. Other proteins are secreted to influence the behavior of other cells. Lasker laureate Günter Blobel showed that exported proteins are inserted into the ER as they are translated. Once inside, they are bound by the factory workers of the ER, which are proteins called chaperones. Like Ford's welders, the chaperones help the exported proteins to fold into their final three-dimensional structures. The ER assembly line must perform perfectly. Mistakes are not tolerated. Unlike the auto industry, in biology, there are no recalls. Just like Ford's assembly line, Nature's assembly line has an inspector. When Nature's inspector finds defective proteins, it sends a signal to the cell's control room, which is the nucleus. Just like a factory control room, the nucleus dispatches agents that enter the endoplasmic reticulum to correct the problem. The cell's agents are additional chaperones, and their work is essential. If the problem is not corrected, the cell's secretory factory becomes congested and the cell dies. Even worse, if defective products escape the inspector and reach the circulation, they are recognized by the immune system as foreign, and this elicits an inflammatory response. Peter Walter and Kazutoshi Mori, our two basic science awardees, discovered the inspector of the endoplasmic reticulum. They showed how the inspector detects abnormal products and how it signals the nucleus. The mechanism is called the unfolded protein response. It is mechanistically unprecedented. It is conserved in all cells, and it is essential to life. Mori was introduced to the protein folding problem as a postdoctoral fellow with Mary Jane Gething and Joe Sambrook at UT Southwestern Medical School in Dallas. I mention this because one of last year's laureates, Thomas Sudoff, also trained as a postdoctoral fellow at UT Southwestern. Not bad for a standalone medical school in a red state without a coast or a mountain. Maury's co-recipient, Peter Walter, did not train in Dallas, but he did train in the US. He trained with Gunter Blobel at the Rockefeller University right here in New York, which has a coast but no mountain. I mention these facts because they show how the US has been the endoplasmic reticulum of world science. Like newly made proteins, nascent scientists are endowed by their genes with the potential for higher function. Like proteins, newborn scientists need chaperones to help them fold into the tough structure they will need for survival in the harsh external world. Until recently, the US has had the best scientific assembly lines in the world. We imported the best raw material and turned out the best products. Now our scientific factories are threatened by short-sighted cutbacks in federal funding. There are people in today's audience who are trying to reverse this downslide. Let's hope they get the support they need. But I digress. Back to Kazutoshi Mori. He approached the unfolded protein response backward. He didn't start with the first event, the sensor. Instead, he started with the last event, the signal that turns on the production of chaperones when the sensor is activated. From there, Mori and Walter worked backwards, eventually finding the sensor that detects the unfolded proteins and initiates the signal. Mori made his major discoveries after he returned to Japan, where he worked at Kyoto University, mountain but no coast. Walter remained in the US at the University of California, San Francisco, which has a coast and a mountain. The two scientists worked independently in what appears to be a friendly competition, if such a thing ever exists. One of them would find a new link in the signaling cascade, and the other would confirm the finding and use the information to discover the next link. They both started by studying yeast, where powerful genetics allows a rapid dissection of biochemical processes. Soon they moved to animal cells, where the process was similar in principle but more complex in detail. I won't burden you with the names of the proteins and the functions that Walter and Maury discovered. They are listed in the fine articles that have been distributed to you today by Evie Strauss in the official program. Evie, by the way, a really outstanding writer. As well as in the pieces in Cell, Nature Medicine, and the JCI. But I will describe one aspect that is biologically unprecedented. When unfolded proteins accumulate in the ER, they bind to a sensor protein whose sensing domain projects into the ER lumen and whose active domain pokes through the ER membrane to function on the cytosolic side. When unfolded proteins bind to the sensor, they activate the cytosolic domain so that it becomes an active enzyme. The novel discovery is that the enzyme is a nuclease that excises a segment from a specific messenger RNA. Splicing activates the mRNA, allowing it to produce a transcription factor that enters the nucleus and activates the genes encoding chaperone proteins. The chaperones enter the ER and fix the folding problem. Before the Maury-Walter discoveries, the mRNA splicing was believed to occur only in the nucleus. There was no precedent for the notion that such a reaction could take place on the surface of the ER and that it could activate the translation of a specific protein. Mother Nature is very frugal. She never wastes a good idea. It seems likely that other regulatory pathways must use a similar mechanism, but so far they remain undiscovered. In humans, the unfolded protein response is essential in cells that secrete large amounts of proteins. Foremost are the antibody-secreting cells of the immune system, whose secretions are crucial for our defense against bacteria and viruses. The liver also secretes lots of proteins that must be folded properly. A mutation in one secreted protein, alpha-1 antitrypsin, causes the protein to misfold and puts enormous stress on the unfolded protein response. Over time, the control room cannot keep up, and the patients develop liver failure. The discoveries of Walter and Maury are among the most profound and original in all cell biology over the past several decades. They are worthy recipients of the 2014 Albert Lasker Basic Science Award.
Thank you, Mike, for so vividly teaching us about the magnificent unfolded protein response and for reminding us how important funding is if we're to continue to make such fundamental biological discoveries. So Doctors Maury and Walter, if you would come up to the podium to accept your award. To Kazutoshi Maury and Peter Walter, the 2014 Albert Lasker Basic Medical Research Award is given for discoveries concerning the unfolded protein response, an intracellular quality control system that detects harmful misfolded proteins in the endoplasmic reticulum and signals the nucleus to carry out corrective measures. Congratulations. Mr. Overlap? Okay, smile for the camera. Thank you. I'll take that back and hold it for you. Thank you. And Dr. Maury will start us with some comments.
It is my very great pleasure and honor to receive Lasker Award. Looking back, I can point to many happy meetings that changed the direction of my career. In particular, Three meetings come to my mind. The first meeting took me to the United States. I did not like biology in high school. Textbooks seemed descriptive. I felt I had to learn everything by heart. So as a freshman of Kyoto University, most of my learning came from newspaper, where I met molecular biology. I was amazed by the fact that genetic code is conserved from E. coli to humans, which allows scientists to produce human protein in E. coli. I hoped for a bright future in biology, so I switched my major from chemistry to biology. Because since there was no molecular biology program in my school, I studied biochemistry. And after graduating, I had a permanent position at a local university. I worked hard, but I could not enjoy the biochemical project I pursued. I want to do something more important and interesting. So I decided to quit that job and go to United States, learn molecular biology. So in 1989, at the University of Texas Southwestern Medical Center, I finally met the discipline of molecular biology under the mentorship of Drs. Mary Jane Gessing and Joe Sandberg. And there in Dallas, I was introduced to the unfolded protein response, UPR. This is my second happy meeting. I succeeded in cloning IR11, which turned out to be a key molecule to understand UPR in yeast cells. To my surprise, Peter Walter got same molecule at the same time. So in our first paper, published nearly back-to-back in 1993, Peter and I showed that IR11 has a strong kinase activity. And 4.5 years at Texas, I came back to Japan and obtained a position at HSP Research Institute. My next target was a transcription factor specific to yeast UPR. But my new director, Takashi Yura, advised me, be careful. Because IRE1 is a kinase, he said, there may be a downstream kinase cascade. So you may end up obtaining many kinases, but not transcription factor. Because the institute focused on transcription, he told me to think about a method of identifying the transcription factor directly. But it was a difficult task. After 14 months of struggle, I finally came up with a simple method called one-hybrid screening, which turned out to be a right approach. It worked magnificently, and in 1996, I obtained HAC1, key transcription factor specific to the UPR in yeast cells. And with this simple one-hybrid screening, few years later, my colleague and I obtained the tumor transcription factor crucial for the UPR in human and mammalian cells. Had I stayed in the United States after my Texas days, I would have probably employed more complicated American-style screening system that would have never allowed me to keep up with a major player like a Peter Walter. The development of simple one-hybrid screening is my third happy meetings. To me, it was a Japanese version of American dreams. So thank you again for honoring me with Lasker Award.
We are so happy you had those happy meetings. And now some comments from Dr. Walter.
Well, thank you so much. 4 years ago, Joe Goldstein issued the ultimate instructions to us on how to win a Lasker. In his short piece in Nature Medicine, he presents a list of advice culled from the experts, as he puts it, and then promptly dismisses it as a not foolproof formula. He continues to quote, as better advice, the British mathematician Godfrey Hardy, who defined in the early 1900s scientific beauty as an art form in which outstanding science gives you cerebral chills and intellectual kicks that combine the qualities of significance, generality, and unexpectedness. Biology, however, is not mathematics, nor is it physics, nor chemistry. As biologists, we are not free to impose our own axioms, to inject beauty into our work, or even assume elegant adherence to deterministic logic. In biology, Mother Nature presents the playing field, and it's our task to decipher how it works. Disconcertingly, nature deploys the strategy of random walk, of mutation and selection, leading to the evolution of the world that surrounds us. She then presents us with the most fascinating puzzles to decipher, the inherently unpredictable Rube Goldberg machines that make up the living cell. When we began working on the unfolded protein response that we celebrate here today, we found ourselves traveling along a road that seemed comfortable and predictable, perhaps too narrow and straight. We asked but simple questions. How does one part of the cell know what's happening in another? And we got a simple answer. A kinase receptor that, like many others, controls the gene expression system. But suddenly the ground shifted, and we stepped into a morass in which the seemingly familiar dots that before gave us a sense of security no longer connected. Walking on, undeterred, we deciphered one of the most unusual cell internal communication pathways, made up almost entirely from an unprecedented potpourri of repurposed components. To top it off, the salient features of what we learned from simple single-celled brewer's yeast hold true for our own cells. And these features now emerge as impacting players in a plethora of human diseases. And this gives us hope that our esoteric findings will be translated one day into tangible benefits for mankind. My main point here is that none of this was predictable. Neither I nor Kazu stand here today as genius mathematicians akin to Hardy, or as artists who begin with an empty canvas and control over the elegance of their work. We are explorers, not designers, facing the chaotic randomness of evolution. Over the last 20 years, we diligently deciphered one of nature's guarded mysteries, and only then could we condense our findings to their most elementary beauty. To us, this has been a fantastic journey, filled with adventure, cerebral chills, and intellectual kicks. Yes, this journey required radical thinking and fearless experimentation. But only in the end did it all combine, serendipitously, into a story of unexpectedness and, we hope, lasting significance and generality. We are thrilled to be here today, and we deeply, deeply appreciate that our work has been so well received. Thank you all for such a wonderful, nice celebration, and I would like to offer a toast to discovery.
So we will now move on to the Lasker DeBakey Clinical Award, and to introduce our awardees is Greg Petsko, who's been on the jury for 11 years, but probably more important to all of you in the audience, Greg is a new New Yorker. He is the spouse of Dr. Laurie Glimcher, who is now the dean at Cornell Weill Medical School. Greg?
Thank you. I'm still a Red Sox fan, however. In 1972, the novelist Michael Crichton, whose Harvard Medical School education seems to have primarily done nothing except turn him into a techno-Luddite, wrote a novel called The Terminal Man. It told the story of Harry Benson, who has electrodes implanted into his brain to suppress his psychomotor epileptic seizures. The electrodes are controlled by a small computer with a power pack under the skin in his shoulder. Unfortunately, this being a Michael Crichton novel, the technology is flawed, Benson is a psychotic, and, well, you can probably guess how this turns out. That same year, in 1972, a young medical doctor in France named Alim Louis Benabid became a staff neurosurgeon at Joseph Fourier University in Grenoble, while also beginning his studies for an eventual doctorate in physics. And also in 1972, in the United States, another doctor, Malin Delong, was finishing up his time as a research associate in the National Institute of Mental Health. Eventually, the work of these two men would coalesce into something strikingly similar, sort of, to the device in The Terminal Man. But its use Michael Crichton could never have dreamed up: a new therapy for the symptoms of Parkinson's disease. Parkinson's disease is the body snatcher. It starts innocently enough, often with just a tremor in one arm. But then, as the cells in the brain that produce the neurotransmitter dopamine continue to degenerate, it progresses, as relentless as a curse. Slowed movement, postural instability, difficulty in walking, and eventually wheelchair or bed confinement and death. More than 1 million people in the United States suffer from Parkinson's, a number that will grow to almost 3 million by 2050. That's more than the population of the city of Chicago. In the 1950s, it had been observed that destroying certain regions of the brain could help treat some movement disorders, including Parkinson's, symptomatically. But it was impossible to prevent concomitant brain damage in many cases, so this approach fell out of favor when dopamine replacement therapy was introduced in the 1960s, which, by the way, was recognized in 1969 with the Lasker Clinical Award to George Kotsias. Symptomatic relief can be achieved for some time with dopamine replacement therapy, but eventually it becomes impossible to administer enough, and the high concentration of the drug produces a complication called dyskinesia, marked by involuntary writhing movements, a side effect that for many patients is actually worse than the symptoms of the disease. Until the work of our clinical award winners, Drs. Benabid and DeLong, once dopamine treatments ceased to be effective, Parkinson's patients were doomed to progressive disability. But they changed that. When Malin DeLong arrived at NIH, he decided to work on a little-studied part of the brain called the basal ganglia. He was still studying the basal ganglia in the early 1980s when a group of drug addicts in California tried to synthesize heroin, missed, and instead made a substance called MPTP, which upon administration immediately threw them into full-blown Parkinson's disease. Scientists quickly used MPT to make a monkey model of Parkinson's disease. That monkey model allowed DeLong to test his idea that the symptoms of Parkinson's following the loss of dopamine were due to excessive and abnormal activity in the output cells of the basal ganglia, specifically a small region called the subthalamic nucleus. He and his co-workers used the new MPTP monkey model of Parkinson's disease and inactivated the subthalamic nucleus with a lesion. When the lesion was made on one side of the basal ganglia, the tremor and muscular rigidity of the monkeys disappeared on the opposite side of the body. So here then was a new approach to symptomatic relief for Parkinson's: inactivate specific regions of the basal ganglia. The question was how to do that without collateral damage. Around the same time, thousands of miles away in his laboratory in Grenoble, Alim Louis Benabid was making a remarkable discovery of his own. He'd been doing neurosurgery lesions on movement disorder patients who were unresponsive to drug therapy, and one day in 1987, he was about to operate on a person with a severe tremor. The standard procedure in those days was first to stimulate the area you were about to destroy, in this case a region called the thalamus, with a 50 hertz electrical pulse to locate the target region and see what would happen when you destroyed it. Since the electrical stimulation itself sometimes seemed to quiet down the tremor a bit during the procedure, Dr. Benabid wondered what would happen if instead he used a different frequency. At just under 100 hertz, the patient's tremor completely stopped. When he turned the current off, it returned. Here, then, was a way to treat one of the symptoms of a movement disorder without destroying any brain tissue. The question was, for Parkinson's disease, which was the right region to stimulate to relieve the most serious symptoms? But that question, of course, had been answered by the work of Dr. DeLong, the subthalamic nucleus. In 1995, Benabid reported that bilateral high-frequency stimulation of just that region in 3 people with severe Parkinson's suppressed their slowness of movement and their muscular rigidity. Further studies showed a long-lasting benefit. Deep brain stimulation, as it became known, actually restored their motor skills, suppressed their tremors, and dramatically improved their ability to conduct normal activities. Deep brain stimulation of the subthalamic nucleus was approved by the FDA as a treatment for the symptoms of Parkinson's disease in 2002. More than 100,000 patients now have had electrodes surgically implanted into that region and typically undergo continuous high-frequency stimulation without adverse effects. If you ever see before and after movies of such people, the results will take your breath away. Before, many could neither walk, write, nor speak normally. After, you sometimes can't even tell there's anything wrong with them. In Act 5 of Shakespeare's Macbeth, Macbeth asks the doctor, "Canst thou not minister to a mind diseased?" And all the doctor can offer is, therein the patient must minister to himself. For 500 years, medicine has had little better in the way of answer for depression, psychoses, motor neuron diseases, dementia, and of course, movement disorders. But now, thousands of patients have electrodes implanted into their brains, connected to a power pack under the skin. But unlike Michael Crichton's Terminal Man, unlike Harry Benson, these patients experience not a tragedy of technology, but a miracle of medicine. What was science fiction has become science fact. What was a story of horror is now a story of hope. And while they await the next breakthrough, one that will completely conquer this disease, patients who had been facing years of relentless deterioration and disability are able to function almost normally, thanks to the work of this year's Lasker DeBakey Clinical Medical Research Award winners, Drs. Alim Louis Benabid and Melon DeLong.
To Alim Louis Benabid and Melon DeLong, the 2014 Lasker DeBakey Clinical Medical Research Award is given for the development of deep brain stimulation of the subthalamic nucleus, a surgical technique that reduces tremors and restores motor function in patients with advanced Parkinson's disease. Dr. Benabid will speak first.
Well, ladies and gentlemen, Mrs. Pomeroy, I would like first to apologize for being not a Native American English speaker, and also this being aggravated by my French accent, and as you can see, some additional effect of emotion. This being said, I was born in France during World War II, then raised as a teenager during the Algerian War. And my brothers and I were taught by our parents that peace was the most important things to pursue. My father was an MD. He wanted me to be a physician. My mother was a nurse. She wanted me to be a physicist. And to preserve peace at home, I had no other choice than being both. I would say then that my life was a science fiction novel. My story was based on the patient's problems and expectations, on the use of increasingly sophisticated tools to win the battle, to the thrill of fighting challenges, and the delight of solving some of them with sometimes benefits for the patients and happy ends, and sometimes not. Every chapter has been an adventure shared with mentors, friends, students. The tempo was made by the goals to reach, the deadlines to meet, and the odds to overcome. Everyone needs to know how worthwhile are his achievements. The first and most important one, the most important rewards come from the patients. Recognition and prizes are the milestones of a career. Some, such as the Lasker Award, are unreachable horizons. You just dare to dream of, but you never believe that you will be seeing them becoming true. Becoming nominated for the Lasker Award by such illustrious members and peers is by itself a real honor. Then being finally awarded is itself an honor, but also an incredible shock, and the honor of this prize is even increased by the fact I share it with a friend and a colleague, a scientist whom I admire and with whom I spent a lot of time along our common careers, such as Malone DeLong, as I said. I thank you all for your trust and your belief and acknowledgement in what I tried to achieve. I thank you.
I want to say a few words about how I got into science, but I'm only going to say that I've just been very curious about things most of my life, which has gotten me in trouble more than a few times. And I'll leave those for after dinner. But I acquired a growing fascination at the university in understanding behavior and brain and behavior, how brain controls behavior. And it was really at the NIH in the early or late 60s that I began really to do something serious in research. I had a key position, or a great position, in a research lab that was pioneering work on understanding correlation between behavior, that is movement, and single-cell activity in primates. This was an opportunity of a lifetime. I got there a little late, and to find that most of the choice regions of the brain to study were already assigned to other fellows. So I had the basal ganglia as leftovers, so to speak. But it turned out to have been probably the best thing could have happened. I was asked, as others were, to figure out what those parts of the brain had to do with the control of movement by correlating activity with behavior. For many years, I thought only of understanding that and had not really thought much about finding a treatment or a cure. Those were not the driving, was not the driving force, but was really focusing on anatomical organization, connectivity, networks, and what these cells were related to. And I'm not going to talk about that much at all because it was really almost 2 decades later when this opportunity arose, which you heard about with the MPTP model of Parkinson's in primates that was discovered quite unexpectedly. So it became possible to actually study the parts of the brain that were involved with Parkinson's in a live, moving animal. And this basic work that we had done was key, I think, to really unraveling and understanding what was going on in the Parkinson model. We immediately found abnormal activity. There were those who said, you know, this is foolhardy, that you'll not see anything. But we did, and I won't name those who said that. But, and we found quite, I say unexpectedly, but also expectedly, that when we interrupted that activity by creating a small lesion, and we're talking lesions that are millimeters in diameter, tiny part of the brain, that this immediately, within minutes, alleviated the tremor, rigidity, and animals began moving, as you heard, on the other side of the body. I think the most important part of that really was that we had new insight into what might be causing these disturbances, and also a strong rationale for surgical approaches, which, as you heard, have been largely abandoned for all of that time since the introduction of levodopa. And it also provided an effective target then specifically for treating Parkinson's disease. And this, of course, proved to be most clear with the introduction of Alan Benabid's technique of high-frequency stimulation, which targeted the subthalamic nucleus. Well, the Lasker Awards target individuals, but I think they also serve to really bring attention to progress and developments in a field that is really, in this case, what we now call neuromodulation. And that word has a lot of different meanings, but I'm using it here to refer to the targeting of specific regions of the brain and really networks, as I'll emphasize in a minute, that allow us to change the activity within the network. It can be done by just making a lesion, that is destroying that part of the network, but stimulation offers enormous advantages, as you know. Neurologists spent a century or more trying to understand brain function by correlating the location of a lesion with the behavioral changes that occurred. And that was, of course, not under the control of the experimenter. That was nature. It was nature's accidents, if you will. But with deep brain stimulation, we're now able to not only treat disorders, but to investigate and understand the effects of activation of brain regions. So this has been a really tremendous advance and opportunity. We used to think about brain centers, and now we talk about networks that connect in a very specific manner different regions of the brain. Understanding, and I think how this focused stimulation and how these disturbances come about is one of the major challenges of our time. And I think it's come to the last chapter here. I think it's worth commenting just on the big picture of how discovery comes about or breakthroughs. And there might be a sense that these come because of concerted, focused effort on something, but I think many times they come about serendipitously and by accident, not by intent, but just when we're trying to understand how things work. And by rushing to find a quick answer, we may miss an opportunity that can only be revealed by really understanding the basic science underpinning that, and then finding a really new treatment. So basic science, I think, is really critical, and what these breakthroughs are really come about after, decades of work, and I'm very grateful for all this recognition, but I want to use it to really illuminate that and all of the people who have contributed to this work. Thank you.
So now we'll move to the Special Achievement Award, and the presentation will be presented by Mark Tessier-Levine, who's been on the jury for 5 years. It's the shortest tenure of those presented us today, but he's the tallest member of the jury. Mark.
Pioneering scientist, social activist, humanitarian, Mary Claire King is also foremost a free spirit who, for over 4 decades, has marched to the beat of her own drummer, animated by the impulse to solve iconic scientific puzzles, a passion for victims of disease and social injustice, and a warm humility that belies her profound impacts on science, on medicine, and on society. Now, perhaps because I'm from Canada, when I think of Mary Claire, the person who comes to mind is Wayne Gretzky. The hockey great with the most career goals and assists, who was a master of the hat trick, scoring 3 or more goals in 50 games. Now, all scientists strive to contribute lasting findings to our body of scientific knowledge, important assists, if you will. Most also aspire to make at least one discovery that changes the way we think, or that has a direct, tangible effect on their fellow humans, to score a goal. Few succeed, of course, in these loftier aims, and even few score more than one goal. Mary Claire is the rare scientist to have scored a hat trick with major impacts in at least 3 fields: genome evolution, medical genetics, and molecular forensics. Like the great one himself, Mary Claire is in a league of her own. Trained in mathematics, Mary Claire began her unconventional trajectory through biological science as a graduate student in statistics at Berkeley in the early 1970s. There, realizing that statistics could help solve problems in genetics, she switched fields, joining the laboratory of Alan Wilson. With a flair for tackling the biggest, most foundational problems, she sought to determine the degree of identity between the genes of humans and chimpanzees. This was long before direct sequencing of DNA made it possible to compare genomes in detail. Even so, she applied statistical analysis to what data she and others could generate on proteins and DNA molecules from the 2 species, and made the startling discovery that they are more than 99% identical, much closer than previously thought. The genetic similarity seemed to contradict anatomical and behavioral distinctions, leading her and Wilson to propose that the marked divergence arose not from markedly divergent DNA sequences, but rather from a small number of mutations affecting gene regulation, a theory at the center of current evolutionary thinking. The ramifications of her findings for how we think about ourselves are profound, and they created a major stir at the time in both the scientific and the lay communities. It's hard to believe that this discovery could be topped, yet that's exactly what she achieved. Returning from teaching abroad, more on this later, Mary Claire accepted a research position to study breast cancer and proceeded to do what she does best. She thought hard and unconventionally about how to make a contribution. Thus began a 17-year odyssey that led her to discover the first gene defect that predisposes to breast cancer. What was revolutionary about her finding is that breast cancer is a common, complex disease, and the prevailing view at the time was that such diseases arise from interactions among multiple genetic and environmental factors that would be extremely difficult to disentangle. But Mary Claire was intrigued by hints that there might be inherited forms of the disease and tested this head-on by studying over 1,500 families in which multiple members were affected by breast cancer. She showed mathematically that clustering of cases could be best explained by the presence of a disease gene in a very small fraction of families. Yet the field remains skeptical, so to convince the skeptics, she set out to determine where the hypothetical gene resides in our DNA, working at the cutting edge to apply tools for gene mapping that were just being developed. She also made the crucial decision to focus on select families in which breast cancer appeared to strike early, as she intuited that there was something different about them, a decision that in retrospect was key to her success. The work culminated in 1990 in her demonstration that in those families, inheritance of a small stretch of DNA conferred a 100-fold greater risk of breast cancer, leading her to predict existence of a susceptibility gene in that region, which she named BRCA1. Her announcement electrified the community, converted even the most hardened skeptics, and set off a race to isolate the affected gene. The isolation of BRCA1 and its close relative BRCA2 made it possible to diagnose whether a woman in an affected family is at high risk of developing breast cancer and for her to seek prophylactic treatment, as recently highlighted by Angelina Jolie. Just as important, the innovative approach Mary Claire took, that is, to focus on specific families with striking inheritance patterns of particular forms of a common disease, was a methodological breakthrough that has since provided a roadmap for isolating genetic forms of other more common diseases. Mary Claire also had her third big win in that same period. She always possessed a strong sense of social justice and had embraced activism, for example, in protesting the war in Vietnam. She had also developed an affinity for Latin America, spending time teaching in Chile after a PhD. There, she witnessed the 1973 coup that overthrew the Allende government and led to incarceration or death of many government supporters, including some of her acquaintances. It's perhaps no coincidence, then, that when approached later, she was moved to help address the problem of identifying the families of young children in Argentina who'd been kidnapped and given to military families during the dictatorship there. In 1984, she reported a first method to help match children with surviving relatives based on a blood test. In doing so, she was a forerunner in the nascent field of molecular forensics. She soon conceived an even more powerful test based on mitochondrial DNA that has been used in matching many more children to relatives and is widely used for identification of human remains, including the previously unknown soldier entombed at Arlington, as well as other soldiers and victims of executions around the world. What made it possible for Mary Claire to score so big in 3 fields? Wayne Gretzky famously said that his success was due to his ability to skate to where the puck is going to be, not where it has been. Mary Claire's farsightedness in entering new fields where she sees big opportunities is likewise one of her secret weapons. Gretzky was also fond of saying that you miss 100% of the shots that you don't take. Mary Claire's drive to keep taking shots on goal, her relentless probing, and her fearlessness in entering new fields also help explain both her past successes and her staying power as she continues to make fundamental contributions to both science and to society, including on the genetics of deafness and schizophrenia, and in fostering interactions between Israeli and Palestinian scientists. Mary Claire King is a role model for how a brilliant scientist who remains curious, courageous, driven, and compassionate can multiply her impact in a truly magnificent way. Science and society are the great beneficiaries of Mary Claire's passion, for which we are all most grateful.
Mark, thank you so much for that tribute to a great scientist and humanitarian. Dr. King, please step forward. To Mary Claire King, the 2014 Lasker Special Achievement Award in Medical Research is given for bold, imaginative, and diverse contributions to medical science and human rights. She discovered the BRCA1 gene locus that causes hereditary breast cancer and deployed DNA strategies that reunite missing persons with their families. Congratulations.
Wayne Gretzky. Genetics makes strange bedfellows. So, one of the many pleasures of being chosen for the Lasker Award is the realization that I'm part of a culture of science and that the community that makes up this culture considers me part of it. It may be that some scientists come to this realization very young. It has taken me far longer. So as a result, I've been musing about this culture, what it means to us, and what we've gotten ourselves into. The central feature of life in science is that we want to be here. We enjoy this life. Science is fun. Genetics is enormous fun. It allows us to be imaginative and creative in an elegant way. It is work for a greater good, yet appeals to our curiosity and our pleasure with puzzles solved. The work is useful and valued by society. What more could we ask? In genetics, each new discovery is opening a gift box from nature. We can understand the pleasure that discovery holds for other geneticists, even other geneticists we never met. For example, my work draws directly on results from Gregor Mendel, Charles Darwin, and Paul Broca. I can see exactly what excited them about patterns, revealed to Mendel by shapes and colors of peas, to Darwin by shapes of bills and shapes of feathers of finches, and to Broca by shapes of the brain and clustering of breast cancers in families of his patients. I wish for them only that they had known each other. In the middle of the 19th century, from the 1840s through the 1870s, Mendel was working in Brno, Darwin in Kent, and Broca in Paris. Broca was an admirer of Darwin's work and a member of a society of free thinkers devoted to the study of evolution, but none of the three met each other. Imagine if they had had Skype. Genetics would have leapt ahead 100 years. 150 years later, it's our extraordinary luck to be working in a revolutionary period in genetics. It's irresistible not to succumb to a sense of unlimited horizons. Every result, regardless of gene, pathway, or organism, is part of a whole story that eventually will make sense to us. The lesson of evolution is that the natural world is ordered and that people can figure it out. Sometimes students ask whether genomics hasn't replaced genetics, whether genetics isn't old hat, displaced by spiffy new technology. It hasn't, and it isn't. Genetics is a way of thinking. Genomics is a set of tools. The questions geneticists ask now are the same as those asked by Mendel, Darwin, and Broca. The questions go to the nature of life and the meaning of being human. What has changed is our capacity to answer them. In genetics, there are difficult questions, and incredibly difficult questions, but we do not acknowledge any unsolvable questions. Genetics as a way of thinking enables us to frame questions in a testable way, while genomics offers the means to carry out the tests. To me, the greatest challenge in tackling new discovery is framing questions in ways that are meaningful both to the scientist and to the larger community. In my efforts to do this, I have been guided by three principles, first suggested to me by Zena Stein, at table 22, about 40 years ago. First, the most important questions come from people on the front lines. Second, the most righteous projects demand the most rigorous science. And third, no question is too big to ask. Thank you very much.
That brings us to our close of the 2014 Lasker Awards ceremony. I thank you all for being here. It's been a terrific day, and one more round of applause for our new laureates.